So we're ready for Chapter 4, a tour of the cell, and we're going to do something a little different this time. You have the ability to download this piece of paper that I'm going to be writing on, and you can fill out your copy as I fill out my copy here. That'll give you something to study by. It'll give you a little study guide when you get done. And we'll try this instead of the PowerPoint for this chapter. So in Chapter 4, we're going to take a quick tour of the cell. Cells, of course, are very small, and so we'll need to use a microscope to look at our cell. Microscopes use light to illuminate specimens. An electron microscope uses electrons to see the specimen. There are two terms that we'll be using: magnification and resolution. Magnification increases an object's size. Frequently in our book, where you see a picture of a cell or part of a cell, on the side you'll see what the magnification is. If it's been magnified 100 times, 1,000 times, so that you can see it. So that's just making it bigger. Resolution is a measure of the clarity, the ability to distinguish two nearby objects as separate. So on the left side here, we have the image of a dog with good resolution. And on the right side, we have a very blurry dog. The dogs are the same size, so they have the same magnification, but the resolution is much better on the left side. And as you can see, magnification without resolution doesn't do us a whole lot of good. So before we go on, let's look at what our microscopes can and can't tell us. The unaided eye can see down to a little less than a micrometer. The light microscopes are generally used from about here down to here. We can see some bacteria with a light microscope, but we can't see the very smallest bacteria. And we can't see viruses at all with a light microscope. An electron microscope, though, can see viruses and small bacteria and even ribosomes and small proteins. An electron microscope generally can't see atoms. There's a special kind of electron microscope that can, but in general we can't see small molecules like water or individual atoms. Now, in our tour of cells, there are two different types of cells. There are eukaryotic cells and there are prokaryotic cells. And they have similarities and differences. All cells have a plasma membrane. This is what separates them from what's inside the cell from what's outside the cell. And it's pretty much an absolute requirement. They all have cytosol, which is a thick liquid inside that membrane. They all have to have chromosomes because they have to be able to pass on genetic information reliably. And they have ribosomes. I don't have room to put ribosomes. We'll put them up here. Ribosomes are what's going to make proteins, the manufacturing hub of the cell. Now to the differences. Eukaryotic cells have internal membranes. So they don't just have a plasma membrane on the outside, they have membranes on the inside, and they have a true nucleus. fact, that's what eukaryotic means. Means true, and karyotic means nucleus. So they have a true nucleus. Prokaryotic cells are smaller than eukaryotic cells, and they're much simpler. They don't have any of the internal membranes, and they have no organelles like mitochondria or chloroplasts. So the examples for eukaryotic cells are protists. Protists are single-cell organisms that are very complex. They tend to be kind of large, and they have internal membranes, they have a nucleus, and they have organelles. Animal cells are all eukaryotic. Plant cells, all eukaryotic, and fungus. The prokaryotic cells include bacteria and archaea. R-C-H-A-E-A. All right, so let's take a close-up look of our prokaryotic cell. This is a typical prokaryotic bacteria. It has flagella, which is used for movement. And we see them down here. Bacteria have a cell wall, which is a rigid structure. Rigid. And it maintains shape. It has cytosol, which is a thick fluid inside. It has ribosomes. These are the structures that assemble proteins. It has chromosomes that carry the genes. And it has a nucleoid, which is where the DNA lives. Note that it does not have a membrane around it, so it's different from the eukaryotic. This is a eukaryotic cell. It's much larger than the prokaryotic cell that we just looked at. It has 4 basic functioning groups. The first is the nucleus and ribosomes, which give us genetic control. It has an endoplasmic membrane system, and this is involved in the manufacture, distribution, breakdown of molecules. It involves the ER, which is the endoplasmic reticulum, lysosomes, peroxisomes, and Golgi apparatus. It involves the energy converting organelles, mitochondria, and chloroplasts. Chloroplasts are found in plants. They're what makes the leaf green, and they're what will absorb energy of the sun and turn it into sugars. The last of the 4 main functioning groups are the cytoskeleton and the cell surface. This provides structural support. And communication between cells. Okay, so let's look at this diagram and compare plant and animal cells. So plant cells have a cell wall. Plant cells have chloroplasts. Chloroplasts, like I just said, allow the plant to harvest energy from the sun and make sugars from it. They're green, and if you took lab, you got to see some chloroplasts move around. They have a special communication channel. It's kind of a very small hole in the side of the cell that it leads to another cell, plasmodia. And plants have vacuoles. This is the compartment that stores stuff. It can store water, or it can store food, or it can store waste. Animals have lysosomes. They help break down things, and they have centrosomes, which is a structural component. Both types of cells have nuclei, or a nucleus. Nuclei is plural. They both have plasma membrane. I'll abbreviate that. They both have endoplasmic reticulum. They both have Golgi apparatus. And something that people sometimes miss is that both plant and animal cells have mitochondria.